All right, this video is going to cover gamete formation, otherwise known as meiosis. They look a lot alike and they sound a lot alike, but they are very, very different. I'm talking about mitosis and meiosis if you didn't get that, sorry. Meiosis is the formation of gametes. So that means that this is how in females we create eggs and in males how you create sperm. Meiosis has two cell divisions, right? In mitosis, you just had the one. In meiosis, you have two cell divisions, but there's only one replication of the chromosomes, right? So in mitosis, we had a replication of chromosomes, but then the cell only divided one time, so that each new cell had a full set of chromosomes. But in meiosis, you have one replication of the chromosomes at the beginning, but then your cell goes through one cell division and then another cell division. So if you think about it mathematically, right, the end point of meiosis is going to be very different than the end point of mitosis. So through meiosis, you are going to produce 4 genetically different cells. So each cell is only going to have half of the information that it needs to form another organism. The cells are going to start an interphase, and it's very similar to the interphase of mitosis. We are going to go through that the DNA replication, and we are going to have the 92 chromosomes, just like in mitosis. In meiosis 1, so this is the first cell division. So in this image, it's just going from one cell into 2. We are going to reduce the chromosome number in half so that each new cell has a full set of chromosomes. The first stage of meiosis 1 is called prophase 1, and this is very similar to mitosis, where the chromosomes are going to form. So we are going to go from chromatin, which is the loose form of DNA, into chromosomes. During prophase 1, we have something called homologous chromosomes. Homologous chromosomes are going to have the same genes located on them, but you are going to have one from mom, one from dad. So the prefix homo means the same. So these chromosomes have the same genes on them. So they are going to come together because we are family, right? And they are going to form a tetrad. Tetra means 4. So when in the image you see that the two X's that are the same size come together, the red one, let's say, is from mom, the blue is from dad. You have 4 sister chromatids there. Each one has the same genes on it. Okay, so the 4 sister chromatids that code for the same genes are going to be called a tetrad. So in this image, this is going to be what your karyotype might look like. You have 2 of each chromosome. You get one from mom, one from dad. And this is really important to realize when we go through genetics, right? When you do one of those genetics crosses, right? You are going to get one letter from mom, one letter from dad, which is going to determine the traits that you have. And that's because of these homologous chromosomes. So again, the same traits are found on each one. So like for instance, in chromosome 1, we might have the gene for hair color, or eye color, or height, or freckles, or hair texture. But each person is going to have a unique combination of the traits, also known as they might have different alleles. So each portion, like let's say the very tippy top of chromosome 1, is going to have the DNA that's going to code for your eye color, but mom might have blue eyes in that region of chromosome number 1, but your dad's DNA might code for brown eyes. So either way, that same portion of chromosome 1 is coding for the same trait, but they can code for different versions of that trait. So this is kind of just an image that you probably already understand, right? You have 2 chromosomes in your normal cells. That is the single lines. So the single green line maybe came from mom. The single purple line maybe came from dad. They code for the same genes, but they might have different versions of that gene on each of them. And through replication, then we get 2 of each. Those are the X's. Something cool that happens during prophase 1 is called crossing over. This is a way to increase the genetic variation in organisms. So what happens is when your homologous chromosomes come together and they form their tetrad, some of the times the arms of the chromosomes can kind of cross over. And when that happens, the arms can break off and then recombine on the separate chromosomes. So this is called genetic recombination. So now the chromosome from mom, let's say that is the red one, now has some of the information from dad on it. So that is going to create a complete different DNA sequence than what your mom has, right? So we aren't actually 50% mom, 50% dad. Due to crossing over, we might be a variation of that. The second phase of meiosis 1 is called metaphase 1, and this is kind of like it is in mitosis where the chromosomes are lining up in the center of the cell, the middle of the cell, but this time they're lining up as tetrads. So we have the homologous chromosomes lining up next to each other in line instead of single file. So now we're lining up in pairs instead of single file. Oh, also, in my metaphase 1, we have the spindle fibers that are coming, and they're attaching to those centromeres again. In anaphase 1, this is where we are going to pull the homologous chromosomes apart. So we're not actually separating the chromosomes, we are separating the homologous chromosomes. Okay, now let me say that again. We are not separating the chromatids of the chromosomes, we are separating the 2 homologous chromosomes. So the chromosome from your mom, that's made up of 2 sister chromatids, might get pulled into one side of the cell, while the chromosome from your dad, made up of 2 sister chromatids, might get pulled to the other side. That's a big difference from anaphase in mitosis. And also the cell is going to start elongating during this process. The next phase is telophase 1. This is very much like mitosis, where each pole now has a diploid set of chromosomes, and it's going to kind of untangle itself, unwind back into chromatin, and you are going to have those nuclei reform. And then cytokinesis happens where the complete separation of the cells happens, and you get 2 diploid daughter cells. Now, in between meiosis 1 and meiosis 2, there is no interphase. So these cells are not going to replicate their DNA. They're not going to grow in size. They are just simply going to then enter into meiosis 2, which is extremely similar to mitosis. Now these have the same exact steps. We have prophase, metaphase, anaphase, and telophase, but in meiosis 1, they were prophase 1, metaphase 1, anaphase 1, telophase 1. Now in meiosis 2, it's prophase 2, metaphase 2, etc., etc. So in prophase 2, we do not have the replication of chromosomes, right? But we are going to have the nucleus break down, and we are going to have the chromosomes form. So this is exactly the same as prophase in mitosis. In metaphase 2, again, this is exactly identical to metaphase in mitosis. We are going to have the chromosomes line up single file in the middle of the cell. And these spindle fibers at both of the poles are going to attach themselves to the centromere of each chromosome. In anaphase 2, this is exactly the same as mitosis. We are going to pull the sister chromatids apart in each of the cells. The cell is also going to elongate, in case you care. And then in telophase 2, again, same as mitosis. The nuclei are going to reform, and then the chromosomes are going to unwind into chromatin. And then we have a second cytokinesis, which is going to separate each cell into 2 more cells, making 4 total haploid cells, which are known as gametes, because they only have half the information you need to make a human, or an organism, I should say. So that's why it takes mom and dad in order to make a baby. It for meiosis. So the biggest differences between mitosis and meiosis is mitosis is going to go through 1 cell division, and it's going to produce 2 identical cells. In meiosis, you have 2 cell divisions, otherwise known as you're going through cytokinesis twice, and you are creating 4 haploid cells. So 4 cells with only half the amount of DNA that you need. So those are the 2 biggest differences. You will need to know that for your test. I hope these videos helped. I'm sorry if I sound like a robot. It's my cheap computer. It's not me. But yeah, hope it helped.